and in Tel Aviv Medical Center in Israel. Unfortunately, I could not attend the meeting and be with you physically, but my student, Guy Gurevich, I hope he is raising his hand, is in the hall and will be able to answer questions later on. I will present our work using mesolimbic neurofeedback for mental and physical health. The mesolimbic system is composed of multiple regions, cortical and subcortical, that are heavily involved in different aspects of reward processing, consumption, as well as incentive processes. I will focus in my talk on two core regions in this system, the nucleus accumbens, which is part of the ventral-striatum, marked here in green, and the ventral tegmental area marked here in red. These regions luckily are well depicted by fMRI, by functional MRI, as seen here in two examples from our studies, but this has been demonstrated in accumulated work throughout the years. On the left, you see evidence for incentive processes in VTA and ventral-striatum in a task of approach under risk. And on the right, you see hedonic processes in the ventral-striatum when people were listening to pleasurable music. Interestingly enough, these processes and the mesolimbic system was also shown to be disturbed in different health conditions. In mental health, it was shown to be disturbed in anhedonia, which is the difficulty to enjoy and express positive feelings, apathy, difficulty to initiate behavior towards positive goals, addiction, and even acute stress. In physical health, it was shown to be related to immunity function, inflammation, and pain analgesic. So it's a very important system for our health. This led us to aim to try and augment health through mesolimbic modulation using neurofeedback. And we based our assumptions on two examples, which I will further show you in this talk. So the first example related to anhedonia, and there are evidence, accumulating evidence that in patients with that suffer from anhedonia, the ventral striatum is less activated to reward the stimuli relative to healthy controls. And this kind of lead to simple assumptions that if we upregulate the ventral striatum, we will also enhance hedonic processing and possibly also improve depressive condition. And on the physical side, we have evidence, very nice evidence from animal models of rodents showing in a causal manner that invasively stimulating the VTA resulted in improved immunity, as shown here, one example in responding to E. coli infection on a dish. But this is invasive, and in humans, we aim to enhance, increase activation of the VTA non-invasively, and hoping to enhance immune function also in humans. So to reach these two goals, we are facing two technological gaps. One is the scalability gaps, and the other one is the invasiveness gap. And I will describe our work on these two sides of the screen. So starting from the scalability, to map the mesolimbic system, the best way to map the mesolimbic system, which is in this case that I'm describing here, a relatively small nuclei in the middle of the brain, we must use the functional MRI. But as you know, functional MRI is not very scalable. It's not accessible and affordable for everyone, and definitely cannot go home. For that, if we want to be able to train people repeatedly and in different situations, we must use more scalable measure like electrical encephalogram, EEG. But EEG basically cannot detect activity deeply in the brain in a small nuclei like the ventral striatum or the VTA. It suffers from poor neuroanatomical precision. So to deal with this to actually take advantage of the two abilities of these methods, we developed an approach we termed EEG fingerprinting of fMRI. And specifically, I will show you how we did it using for the ventral-striatum activity. We brought people to the lab and recorded simultaneously EEG and fMRI. And because we were interested in the reward processing, people were listening to music while we were measuring their brain activity, and specifically pleasurable music that they selected prior to the scanning. Then we extracted features from the fMRI and the EEG signals and introduced it to machine learning algorithms. And this allows us to predict the EEG signal, to predict the fMRI activity from the ventral striatum by the EEG features, specifically frequency-related and temporal-related features. And now we have a model, we call it electrical fingerprint, or EFP. I will use this term from now on, EFP, instead of EEG. And we can calculate it online and have a time course of the EEG that can be used now in the neurofeedback loop. But just, of course, you need to believe me that this was actually related to the ventral striatum in the fMRI, and we did we validated it in different ways and published it. Specifically, there is also publication on the ventral striatum by Zinger et al. But you can see here on the left corner that we brought a new group that was not used for the modeling, and were able to show that indeed the EFP is correlated to fMRI in the ventral striatum, specifically in the location neuroanatomically used for the modeling. So now we have this model, and we can use it in neurofeedback. But before that, we also wanted to validate it process-wise and show that it's actually related to increased pleasureness. And in the fMRI, we were able to show, as others showed before, that indeed this is when in the most pleasurable moments, while people listening to music, we get more activation in the ventral striatum in bold than in the less pleasurable moments. And nicely, we're also able to show that in the validation cohort, when people actually reported on high pleasure, we also get greater signal of the EFP. So now we wanted to introduce it into the neurofeedback loop, and in order to enhance the process probing, we used music as a feedback, and we call it musical neurofeedback. And specifically, we used personally selected music, which is most pleasurable, but we scrambled it, and as you can hear here, it was scrambled at first, and while people were trained to upregulate the ventral striatum, and they were successfully upregulating it, the music became clearer. Okay, I don't have time to continue, of course, but this is very rewarding, and this is what we want. We want to reinforce people with a reward while they are being trained to modulate the brain. And this is a collaboration with Montreal group, Robert Zatora and Alan Degern, together with Nomi Zinger in our lab. So now we have this loop with music and the EFP as the target for modulation. And these are the results from the original group in Montreal. In blue, you see the test group, and in orange, a control group that obtains signal from another person, not their own signal. And you can see that with time, after 6 sessions, people getting better in upregulating the ventral striatum using the EFP signal as a target relative to the orange group, which over time don't get better. And we replicated it in another cohort in Israel, the blue group becoming better. So people becoming better in upregulating, but also there is a learning over time. And we brought the people after the training with the EFP again to the scanner, and were able to show that there is target engagement. People were asked now to upregulate the fMRI signal using real-time fMRI, and you see the test group in blue is better in upregulating than the control group. And this was correlated to how good they were in upregulating the EFP outside the scanner. You see the correlation 0.7 for the blue group. And lastly, we measured using the SHAP anhedonia. Even though this is healthy subjects, there are still a spectrum of hedonic feeling. And after the training, we were able to show that the more they were upregulating, the more the capacity to upregulate was greater, the less anhedonic they reported after the training. So this is actually hopeful that if we now take it to patients, we can improve their anhedonia and improve their depression. And we have initial results showing that we're actually able to do it. And unfortunately, I cannot bring it at this point, and maybe next year. So now this was the scalability gap. We were able to show that in a scalable manner, we can upregulate the mesolimbic system and improve the process. Now, can we also improve the body? And for that, we were relying, as I told you, on studies in animals, but these were invasive studies, and we wanted to see whether we can actually do it non-invasively using neurofeedback in humans. So neurofeedback, in a way, it's our best way nowadays, non-invasively, to demonstrate causally that changing something in the brain is actually resulting in changing something either behaviorally or biologically. So here we brought people to the scanner because we wanted to actually modulate the VTA, which we didn't have the electrical fingerprint for. It's a very small region. And we asked people to we trained people to upregulate the VTA as well as the nucleus accumbens together 4 times in the scanner. And after the 4th time, they were vaccinated for hepatitis B. This way, we were manipulated, challenged the immune system, and we wanted to see whether we can actually demonstrate improved immunity to the vaccination. And we assessed the antibody specifically for hepatitis B 14 days and 28 days after the end of the training. And importantly, we assess the success of the upregulation in the brain, of course, using the fMRI, looking at the VTA and the nucleus accumbens specifically in control regions. But we also assess the mental characteristic of this training. We ask people, what did they do while they were upregulating the mesolimbic system in order to tap into the mental aspect of this capability. And this is the results. We measured the VTA and the nucleus accumbens. I show you the results of the VTA because this was our main assumption based on the animal work. And you can see that the experimental group and the control group both upregulated the VTA against our assumption that we were hoping to see more regulation in the experimental group. But our explanation for this is that the neurofeedback is actually a reward task by itself, and upregulating the mesolimbic system is a domain general process of neurofeedback. So in order to look for the immunity after the vaccination, we combine the groups, because we realized both groups actually learned to upregulate their VTA. And we ask, is it related to the antibodies that we obtained from the blood sample? And you can see here the correlation on 60 subjects altogether. The more they were upregulating the VTA, the more we measured immunoglobulin for hepatitis B in the blood. So we actually demonstrated the relationship between modulation of the mesolimbic system and change in the immunity function immediately after. Lastly, we ask, is this related to a specific mental strategy that people were actually activating while trying to upregulate the mesolimbic? And we gave people different features mentally to tell us whether they were using it or not. And the most significant feature, to our surprise, but according to our expectation, was positive expectations. And you can see this is more evident when VTA was highly activated relatively to low activated, high activated in dark blue and less activated in light blue. And this was not found neither for the nucleus accumbens nor for the control regions. Positive expectation, as we know, are strongly related to placebo, and we assume that we actually target through the neurofeedback this mechanism that might help physical health through activation of the immune system. And this was also the assumptions in the animal work, but now we demonstrated it in humans. So to summarize, I hope I show you how we can address translational gap for reward activation for physical health and mental health, using upregulation of ventral-striatum for mental health and upregulation of the VTA for physical health. And of course, this is not the only regions in the brain that are might related to either mental or physical health. This is just an example. We hope to actually probe other regions like cognitive control regions and emotional regulation system. And this is now being advanced as a therapeutic mechanism by Gray Metals Health, which I have to disclose. I am their chief medical scientist. Thank you for listening, and enjoy the rest of the symposium.